Okay, so problem 423 is kind of a continuation of problem 422, but it's with a little bit of a tweak. If you draw out the epistatic pathway down here, if you remember in 422, you had the big C allele that came here and blocked the activity of the big A allele, the product of the big A allele. And so in this one, though, it just blocks this second pathway from gray to black, the activity of the big B enzyme. Okay, so that's the only difference between the two problems. You have the same mating. You have a, big A, little b, big C, mating with little a, big B, little c. In the F1s, you get a trihybrid, right? And the trihybrids cross with each other. So here, this is the setup cross. Here's the pathway. The best way to do it then is to say, okay, let's think about what genotype would give us a black individual. And so you say, okay, you have to have the big A allele, the enzyme that results from that big A allele being transcribed and then translated to convert the white to gray. You then need the big B to convert from gray to black, but you can't have the big C because that's going to block that activity. So you have to have a big A dash, big B dash, little c, little c. And so here are these three different events, and let's just look at, okay, to get that black, if you cross big A little a with big A little a, what portion of those offspring are going to be big A dash? That's 3/4. Same with B, big B dash, that's 3/4. And then little c, when you're crossing big C, little c, big C, little c, you're going to get and that cross 1/4 of the time, or 1/4 of the offspring are going to be little c. So all three of these events have to happen, this event and that event and that event. You times those three together and you get 9/16. Okay, so for the second phenotype, white, what do you need to get white? And this one is really easy because you see that anything that has the little a is going to be white, regardless of what's happening at the B gene or at the C gene. Here is your epistasis. Whatever happens at the white gene overrides all of the others. And so to get a white, all you need is a little a, little a. So you're just doing this very simple cross, big A, little a, big A, little a. What portion of the offspring are going to be little a, and therefore have the white phenotype? 1/4, or just to make it consistent, we'll just convert that 1/4 to 16/64. So 9/64 black, 16/64 white, and then we know that the rest of them then have to be gray. So we can just do 1 minus 9/64 minus 16/64 equals 39/64. Another way of doing that for the gray is you could spell out through the pathway exactly which genotypes are going to give you gray. So big A, big B dash, big C dash will give you gray, right? Because you'll have this enzyme to convert to gray, but then the C will block it. You have big A dash, and then a little b would keep it here at gray, and then you could have the little c, which would also make it so that either let's see. Oh yeah, little b, then little c, or little b, then big C, whatever. Either of those are going to not allow you to go from the gray to the black. And so then you can just times out each of these individually. You get 27/64, 3/64, and 9/64. Add those up together, all those together, and you get 39/64. So that's sort of a little bit different way if you could do that, just sort of explicitly spelling them all out, or you could just one minus the other two results. Okay, so that's that one.